What if everything we thought we knew about the origin of our species was only half the story? For centuries, scientists, explorers, and philosophers have asked one question that echoes through every culture and every continent. Where do we come from? We've looked to bones buried in ancient soils, traced footprints across deserts, mapped tools scattered through caves, all in an effort to piece together the great puzzle of the first humans. And while fossils gave us a framework, the truth is, much of what we believed was built on fragments and guesswork. Then came a revolution, one made not of stone or bronze, but of code. In the last 2 decades, a breakthrough in technology has allowed scientists to extract ancient DNA from fossilized bones, sediments, and even cave dust. With it, we began to read the genetic memories of the dead, unlocking secrets once thought lost forever. Suddenly, we weren't just imagining our ancestors. We were listening to them, and what they told us. It was nothing like the story we thought we knew. Ancient DNA revealed that humanity's origin wasn't a straight line. It wasn't one tribe, in one place, at one time. Instead, it was a braided stream of different human groups, migrating, mixing, interbreeding, and adapting across thousands of years. But just when we thought the picture was coming into focus, a new layer emerged. A discovery that linked the smallest molecules inside our cells with the vastness of the cosmos itself. This is where 3-IDATLAS enters the story. Whether you interpret it as a groundbreaking genomic mapping system or a reference to interstellar data influencing ancient timelines, Atlas represents a shift, a widening of the lens through which we view the birth of our species. Imagine this: particles from space, ancient isotopes, or even cosmic radiation patterns helping us better understand when, and maybe even where, the first humans emerged. It sounds like science fiction, but it's grounded in cutting-edge research. Combined with ancient DNA, the ATLAS discoveries didn't just add details. They rewrote the early chapters of our story. We now know that modern humans carry fragments of DNA from at least 3 other ancient hominin groups: Neanderthals, Denisovans, and a mysterious third lineage with no known fossil record. These were not just footnotes in our past, they were family. And yet, they were also alien to us, genetically distant, behaviorally distinct. But through the mingling of bloodlines, they became part of us. Their DNA flows in the veins of billions alive today. So what does that mean? It means our search for the first human is a flawed question. There wasn't a single first. There were many beginnings, all colliding, combining, and collapsing into what we now call Homo sapiens. It means that we didn't emerge, we unfolded, not as one point of light, but as a constellation of early human groups spread across Africa, Asia, and beyond, some of whom we never knew existed until the genes of their descendants whispered back through time. And it means that the origin of our species is not limited to the earth beneath our feet. In some strange, poetic way, through cosmic particles, isotopic fingerprints, or perhaps the radiation patterns of ancient stars, the universe itself left clues about us. This is the story of how ancient DNA and the discovery framework, known as 3I, Atlas, gave us the clearest view yet of where we come from. It is not a story of them and us. It is a story of we, a human family, complex, blended, and beautifully unfinished. So if you've ever looked up at the stars and wondered who the first humans really were, this is your answer. Let's begin. For most of human history, the only clues to our past came from what the earth chose to preserve: bones, stone tools, charred remains of fires, and fragments of ancient shelters. These artifacts built the foundation of paleoanthropology, the science of studying ancient humans. And from the beginning, the picture was incomplete. In the 19th and 20th centuries, as fossil discoveries spread across Europe, Asia, and Africa, a rough story began to form. Humans evolved in Africa and gradually spread across the world. Sites like Olduvai Gorge in Tanzania, Zhoukoudian in China, and the Neander Valley in Germany became legendary in shaping our understanding of early human life. But fossils were rare. Sometimes only a tooth, a jaw, or part of a skull would be found. Entire species were defined from a handful of bones. This scarcity created fierce debates. Did humans evolve in one place and migrate outward? Or did they evolve in multiple places at once, with interbreeding along the way. The two dominant theories, out of Africa and multi-regional evolution, battled for scientific supremacy. The out of Africa theory, supported by fossil dates and later by early genetic studies, argued that Homo sapiens emerged in Africa roughly 300,000 years ago and replaced other hominins, like Neanderthals and Denisovans, as they spread across the globe. The multi-regional hypothesis suggested that modern humans evolved in parallel, in several parts of the world, with continuous gene flow between regional populations. This would mean today's diversity is rooted in deep, local ancestry, not just an African origin. The debate highlighted a deeper problem. The fossil record was patchy ***, biased by preservation conditions, political access, and chance. Ancient DNA changed everything, but before that, our picture of human origins was mostly built on bones, maps, and speculation. As researchers began to discover more hominin fossils in Africa, particularly in Ethiopia, Kenya, and South Africa, the weight of evidence began tipping in favor of the out of Africa model. Sites like Omo Kibish, dating back over 195,000 years, and Jebel Irhoud in Morocco, dated to about 300,000 years ago, began reshaping our timeline for the appearance of early Homo sapiens. These fossils looked recognizably human, with skull shapes and facial features far closer to modern people than Neanderthals or Homo erectus. Yet even these fossils raised new questions. While they were anatomically modern, their brain cases were elongated, not rounded like those of people today. They seemed transitional, not quite us, but close. How many versions of Homo sapiens had there been before the version that took over the world? And then there was the question of Neanderthals. In Europe and parts of Asia, these close relatives of humans thrived for over 300,000 years. They used fire, crafted tools, buried their dead, and possibly even made art. They were not a primitive side branch, they were advanced in their own right. But for a long time, they were seen as entirely separate from modern humans. The assumption was that Homo sapiens either outcompeted them or caused their extinction through war, disease, or displacement. The same went for the Denisovans, an even more mysterious group, identified not by full skeletons, but by a few bone fragments and teeth in a Siberian cave. For a long time, the Denisovans were little more than a name. Their features, behaviors, and even geographic spread were unknown. The only clue we had was their DNA, which didn't match Neanderthals or Homo sapiens, but did appear in the genomes of people in Melanesia, Southeast Asia, and Australia. Before the ancient DNA revolution, scientists relied heavily on typological thinking, trying to categorize hominins based on skull shape, brow ridges, jawlines, and other physical traits. But these physical differences, we now know, can arise from climate, diet, or even random drift, not necessarily from evolutionary divergence. Meanwhile, population movements remain speculative. Archaeologists could infer migration routes from the distribution of tools and bones, but they couldn't prove relationships. Did two hominins with similar tools share ancestry, or did they develop those tools independently? The evidence was too thin to say for sure. And then, just as it seemed we were running out of new bones to analyze, a revolution began, one buried in the microscopic structures inside those bones. It started with mitochondrial DNA, small loops of genetic material passed down from mother to child. Researchers managed to extract mtDNA from Neanderthal fossils and compare it to modern humans. The results showed a clear divergence. Neanderthals were related to us, but they had been on a separate evolutionary path for hundreds of thousands of years. The next breakthrough came with nuclear DNA, the full genomic instruction set that determines everything from height and hair color to immune function and metabolism. Extracting nuclear DNA from ancient fossils was a monumental challenge, requiring extreme care to avoid contamination and cutting-edge technology to amplify degraded fragments. But it worked, and when scientists finally compared Neanderthal nuclear DNA to that of modern humans, they made a shocking discovery. People of European and Asian descent shared 1% to 2% of their DNA with Neanderthals. This meant our ancestors had not merely encountered Neanderthals, they had interbred with them. Neanderthals were not extinct strangers. They were part of our family. This finding flipped the script on the out-of-Africa model. It didn't disprove the theory, but it added complexity. Homo sapiens had emerged in Africa, yes, but as they spread into Europe and Asia, they did not replace other human species completely. Instead, they merged with them. Further research uncovered even more interbreeding events, this time with Denisovans. Populations in Melanesia and Aboriginal Australians carry up to 5% Denisovan DNA. The evidence pointed to multiple encounters, possibly at different times and locations. And then came a third surprise: genetic fragments in modern African genomes that didn't match Neanderthals or Denisovans, suggesting a third, unknown hominin group had also contributed to our DNA. These revelations transformed how we thought about human origins. We were not the product of a clean, linear transition from one species to another. We were the result of mixing, contact, and survival across multiple continents. It also shed light on why fossil evidence had always been so confusing. Traits thought to define modern humans could show up in Neanderthals or Denisovans. Fossils with a blend of features could no longer be dismissed as anomalies. They were likely the result of real gene flow between populations. By the early 2020s, it was clear the story of human origins could no longer be told with bones alone. The genome had become our new fossil record, one that stretched back hundreds of thousands of years and carried the imprints of forgotten encounters, migrations, and adaptations. That's why the development of large-scale genomic initiatives like **3i, Atlas** became so important. Projects like these didn't just look at individual fossils. They compiled enormous datasets from populations around the world, mapping genetic variation on a global scale. They helped scientists trace ancient human lineages across time and geography, detect hidden admixture events, and build complex models of how humans evolved, dispersed, and interbred. What had started as isolated genetic studies in labs became a global effort, a human genomic atlas. As detailed and ambitious as any ancient map carved in stone, the future of anthropology wasn't just in digging up the past. It was in decoding it, one genome at a time. The discovery of ancient DNA was nothing short of revolutionary. For centuries, anthropologists relied on bones, tools, and artifacts to infer human behavior and evolution. These physical records painted a partial picture of our past, but they could never fully explain who our ancestors were, how they interacted, or how closely they were related to one another. That changed in the late 20th century, when a small group of researchers attempted something that many believed was impossible: extracting genetic material from long-dead humans. DNA is a fragile molecule, prone to degradation from heat, humidity, and time. For years, it was assumed that DNA could not survive more than a few thousand years, let alone tens of thousands. But in 1997, scientists successfully extracted fragments of mitochondrial DNA from a Neanderthal bone, found in the Neander Valley in Germany. It was a breakthrough, the first time a genetic connection could be made between modern humans and an extinct relative. Mitochondrial DNA, mtDNA, inherited only through the mother, offered a limited view into ancestry. But it proved one crucial thing: ancient DNA could survive under the right conditions. This breakthrough launched a new field, paleogenomics, the study of ancient genomes. By 2010, researchers at the Max Planck Institute in Germany had gone further. They published the full nuclear genome of a Neanderthal. This time, it wasn't just maternal lineage, it was the full genetic blueprint. And what it revealed shocked the scientific world. The genome comparison showed that all non-African people today carry between 1% and 2% Neanderthal DNA. This meant that interbreeding had occurred between Neanderthals and early Homo sapiens as our ancestors, spread into Europe and Asia. This was more than a footnote, it was a paradigm shift. The idea that our ancestors didn't just encounter, but mated with other hominin species, forced scientists to abandon the outdated replacement model in favor of a more complex picture of human evolution. And the revelations didn't stop there. Around the same time that the Neanderthal genome was being decoded, another groundbreaking discovery occurred: this time in the remote Denisova cave. In the Altai Mountains of Siberia, a tiny fragment of a pinky finger bone, found in 2008, turned out to belong to an entirely unknown group of archaic humans, the Denisovans. When scientists sequenced the DNA from the finger bone, they were stunned. The genome didn't match Neanderthals or modern humans. Instead, it revealed a previously unknown hominin lineage that had lived and interbred with both. Even more astonishing, Denisovan DNA showed up in high percentages in the genomes of modern populations in Papua New Guinea, Melanesia, and parts of Southeast Asia. This meant that Denisovans must have had a broad geographic range, stretching thousands of kilometers beyond the cold Siberian cave, where their bones were first discovered. The revelation of Denisovans opened up a new chapter in human history, one where multiple human species coexisted, migrated, and blended with one another across enormous distances. But how did scientists begin to reconstruct these invisible connections? The answer lay in technological advances. High-throughput sequencing and improved contamination controls made it possible to recover and sequence DNA even from severely degraded bones. Techniques like target enrichment, where only the most important parts of the genome are isolated and copied, helped scientists focus on segments that could yield answers about ancestry, adaptation, and interbreeding. Another breakthrough came with environmental DNA EDNA, genetic material found not in bones, but in the soil of caves. In 2017, scientists showed they could recover hominin DNA from cave sediments, even in places where no human fossils had been found. This expanded the potential of ancient DNA exponentially. It meant that even in the absence of skeletal remains, researchers could identify who had been there and approximately when. These advances led to another startling revelation: the existence of ghost lineages. These are human ancestors whose bones have never been found, but whose DNA still exists inside us today. Genetic signals found in modern African genomes, for example, do not match known Neanderthal or Denisovan lineages. This suggests the presence of other archaic humans, now extinct and unidentified, who also contributed to our genetic heritage. Paleogenomics didn't just redraw the family tree. It restructured it, showing that human evolution wasn't a trunk with a few branches. It was a bush full of tangled, overlapping lineages. The implications reached far beyond ancestry. By analyzing which parts of ancient DNA survived in modern humans and which parts didn't, scientists began to uncover clues about natural selection and adaptation . For example, Neanderthal genes have been linked to immune system function, skin tone, sleep patterns, and even susceptibility to diseases like type 2 diabetes and COVID-19. Denisovan DNA contributed to high-altitude adaptations in Tibetans, allowing them to survive with lower oxygen levels. This showed that ancient interbreeding didn't just add diversity, it added survival advantages. The more genomes that were sequenced, the more complex the picture became. By the mid-2020s, global projects like the Human Origins Program and ancient DNA databases had collected samples from hundreds of sites across Europe, Asia, and Africa. These databases didn't just confirm known migrations, they revealed new ones, including evidence of back migration from Eurasia into Africa, as well as multiple out-of-Africa waves over time. Some studies showed that Homo sapiens may have left Africa as early as 200,000 years ago, far earlier than the traditional 60,000-year estimate. These early migrations may not have been successful in the long term, but they reshaped how we understood the movement of ancient peoples. All of this research built toward a startling realization. There was no single moment when humans became human. Instead, there was a long, gradual process of interaction, evolution, and exchange played out over millennia and across continents. And ancient DNA was the tool that made this visible. What once looked like gaps in the fossil record were now understood as moments of genetic exchange. What had been dismissed as anomalous skulls or tools were now seen as evidence of real hybrid populations. And for every fossil that had been found, DNA opened the door to countless others that had never been preserved. This new vision of human history was not only more accurate, it was more human. It emphasized our shared origins, our interconnectedness, and our ability to adapt and evolve through contact, not just competition. And it set the stage for the next chapter in the story, one shaped by the combined power of paleogenomics and planetary science. Because as much as ancient DNA revealed, it also hinted at something even bigger, that the origins of humans might be tied, not just to the earth we walk on, but to the cosmos above. Until recently, the idea of connecting cosmic phenomena to human evolution might have sounded like science fiction. But with the rise of interdisciplinary research, especially in the fields of genetics, planetary science, and astrophysics, those boundaries are beginning to dissolve. Enter **3i Atlas***. To understand what makes 3i Atlas so significant, we need to break down its two key components. First, 3i refers to the designation of an *** interstellar object ***. Specifically, the third interstellar visitor detected in our solar system after Oumuamua and Borisov. The object was discovered in 2024 and catalogued as 3i Atlas, named after the ATLAS asteroid terrestrial impact last alert system survey that first detected it. This cosmic traveler, 3i ATLAS, wasn't just a hunk of ice or rock. Spectrographic analysis revealed it carried a unique isotopic signature that didn't match anything seen in the solar system. This opened a floodgate of new questions. Could this interstellar material offer clues about the building blocks of life? Could the same particles that shaped planets also have influenced the early environments where humans evolved? While this idea is speculative, it's not without precedent. Many scientists now believe that Earth's early organic chemistry was jump-started by interstellar dust, comets, and meteorites carrying amino acids and other life-forming molecules. If 3i's ATLAS carried similar compounds or traces of exotic elements, it could help calibrate the isotopic timelines we use in geology and archaeology, tightening the dating of early human fossils and tools. More intriguingly, the timing of 3i's ATLAS's discovery coincided with the release of new genomic maps under the name Atlas, short for *** Ancestral Timeline and Lineage Analysis System ***, a massive collaborative project involving genomic labs across Europe, Africa, and Asia. These two Atlas systems, one astronomical, one biological, weren't connected by design, but their overlapping goals revealed something extraordinary. Both were helping to *** trace the origin of humanity *** across time and space. The Atlas Genome Project aimed to build the most complete picture ever of ancient human evolution by sequencing hundreds of ancient DNA samples, from Neanderthal remains to Denisovan teeth to early Homo sapiens in Africa and the Middle East. Researchers began mapping not just migration routes, but ** gene flow ***, showing how populations didn't just move, but ** mixed ***. One of the key breakthroughs was the visualization of these ancient connections on ** temporal spatial genetic maps ***, a kind of interactive atlas showing how human groups split, reunited, or disappeared across millennia. These maps, powered by AI and high-throughput computing, allowed scientists to rewind evolution, almost like watching history in reverse. For the first time, we could actually ** see ** when and where our ancestors merged with Neanderthals, or when Denisovans spread into Southeast Asia. We could trace the arrival of new adaptations like skin, pigmentation genes, or disease resistance markers, and follow them from their point of origin to modern populations. And then, something even more fascinating happened. When researchers began comparing ancient DNA data with ** isotopic timelines calibrated by cosmic input ***, including data from 3i ATLAS, they found subtle but meaningful correlations. Isotopic shifts caused by ancient cosmic events, such as solar flares or meteoric impacts, aligned with moments of accelerated genetic change in ancient populations. Could this be coincidence? Possibly. But some researchers believe we may be entering a new phase of human origins research. One where our *** evolutionary story isn't just written in fossils and genes, but also in stardust ***. In 2025, a group of researchers from the University of Cambridge published a groundbreaking paper titled **Cosmic Catalysts in Human Evolution**. In it, they proposed that spikes in cosmic radiation, detected through isotopic anomalies in ice cores and sediment layers, could have influenced both environmental change and mutation rates in early human populations. What made this theory compelling was that these cosmic spikes appeared to coincide with bursts of genetic diversity in the ancient human genome, especially in mitochondrial DNA lineages associated with early Homo sapiens. The researchers argued that periodic surges of cosmic energy may have increased mutation rates subtly, accelerating adaptive changes or causing genetic bottlenecks that led to the survival of specific traits. Now, enter 3ISATLAS. Although its flyby was brief, it carried isotopic fingerprints never seen before, including variations of heavy hydrogen and exotic carbon that may serve as reference markers. These markers allowed scientists to retroactively reanalyze older geological samples, helping them better date key evolutionary transitions. The real breakthrough came when geneticists layered the newly recalibrated timelines over the Atlas Genome Project's data. Patterns emerged not just in genes, but in timing. For example, mutations linked to immune responses in ancient Eurasian populations appeared to rise around the same periods as cosmic radiation peaks. Could cosmic events have nudged evolution forward? Perhaps not directly, but indirectly by shaping climates, ecosystems, and gene expression? That's becoming increasingly plausible. This fusion of cosmic and genetic data led to the coining of a new term, astrogenomics, the study of how extraterrestrial events may shape terrestrial genomes. It's still a new field, but it's gaining traction, thanks to the insights made possible by 3I, Atlas, and the ancestral mapping technologies now available. Meanwhile, the biological Atlas system continued making waves. Researchers used its algorithms to explore not just ancient lineages, but the depth of interbreeding between humans and other hominin species. By 2027, AI-assisted simulations revealed that Neanderthal Homo sapiens interbreeding likely occurred in multiple episodes spread across tens of thousands of years and at various latitudes, from the Levant to Central Asia, to even parts of Eastern Europe. Even more surprising was the degree to which Neanderthal and Denisovan DNA persisted in regions far from their known fossil range. Genetic traces in the modern genomes of Indian and Tibetan populations hinted at a wider Denisovan dispersal than previously imagined, perhaps all the way west into South Asia, where no Denisovan fossils have ever been found. The Atlas Project's AI models didn't stop there. Using global DNA datasets, they reconstructed lost migrations movements of populations now extinct, whose genetic markers remain only as faint echoes. These models estimated the genetic contribution of these ghost populations and offered hypothetical maps of their dispersal routes, likely habitats, and interactions with other human groups. What emerged from all of this was a stunning realization. Humanity's origin story isn't a tree. It's a ***. Picture multiple threads of human-like species, spreading, intersecting, splitting, and reuniting. Some threads broke off and ended. Others intertwined, strengthening the line. And the Atlas genomic system was the loom that finally let us see the pattern. And yet, even that pattern wasn't fixed. New discoveries, powered by ancient DNA and deep space observation, continued to revise it. Cosmic isotopes helped refine dating of volcanic eruptions, droughts, or mass migrations that once appeared abrupt in the archaeological record. Suddenly, we could correlate environmental disasters or climate shifts with moments of genetic adaptation, like the emergence of skin pigmentation genes or the rise of lactose tolerance in certain populations. This deepened a key insight. Human evolution was not driven solely by biology. It was shaped by interaction with the environment, with other hominins, and possibly even with cosmic forces. To some scientists, this was overwhelming. The Atlas system was generating more data than any human team could process alone. That's why, by 2028, a new AI-driven partner was introduced, LUXE Lineage Understanding and Extrapolation Engine, trained not just on genomes, but on archaeological maps, climate data, and astronomical models. LUX could simulate hundreds of thousands of evolutionary scenarios, iterating over data from both Atlas genome maps and cosmic event logs. It didn't just model human migration, it modeled human potential. And here's where things got really wild. In some scenarios, LUX found correlations between ancient population splits and magnetospheric disturbances sudden changes in Earth's magnetic field triggered by solar events. These disturbances could have disrupted animal migration, rainfall, or even atmospheric radiation, all of which, in theory, could affect human survival, movement, and adaptation. The cosmic connection, once fringe science, was moving closer to the mainstream. The real value of the 3IATLAS discovery wasn't that an interstellar object flew through our solar system. It was that its discovery helped catalyze an entirely new way of thinking about our past, not as isolated branches of evolution, but as a tapestry woven from earthly biology and cosmic timing. The story of the first humans was no longer just a question of bones and blood. It had become a story of genes and galaxies and they were never truly alone. Thanks to ancient DNA, we now know that the blood running through our veins is a shared inheritance, not just from African ancestors, but from Neanderthals, Denisovans, and other forgotten relatives whose stories we are only beginning to tell. Our past is not pure. It is not linear. It is a glorious, complicated mosaic, and it is still unfolding in the not-so-distant past. The origin of humanity was told as a simple tale, a spark of evolution in East Africa, a journey across continents, and the rise of modern humans who outcompeted their primitive relatives. It was a neat, linear narrative, one that fit into textbooks and museum exhibits. But today, thanks to ancient DNA, genome mapping, and the unexpected contributions of interstellar science, that story has been completely rewritten. What we now know is both more complicated and far more beautiful. Humanity did not arise from a single event or a single pair of ancestors. Instead, we emerged through a long and tangled evolutionary process involving multiple hominin species, overlapping migrations, and continuous interbreeding. Homo sapiens were not the chosen ones. We were participants in a vast, dynamic experiment in survival, adaptation, and creativity. The genome, our biological memory, has shown us that our ancestors carried the DNA of Neanderthals, Denisovans, and others whose fossils may never be found. These weren't enemies or evolutionary dead ends. They were part of the same human story, and their genes, still alive in us today, are proof that extinction is not always the end. Through projects like the Atlas Genome Initiative, and with insights triggered by the discovery of 3I Atlas, we've begun to align biological evolution with environmental and even cosmic timelines. We've seen how interbreeding shaped our immune systems, how ancient migrations altered our appearance, and how survival meant adaptation, not just to nature, but to each other. We've also discovered that the concept of a pure human lineage is a myth. The idea that any one population, continent, or race holds a more authentic version of humanity has no basis in genetics. We are all mixtures. We are all mosaics. The true story of human origins is one of connection not separation. What began as a quest to trace a family tree has become a journey to understand the entire forest, full of lost branches, hybrid roots, and overlapping rings of time. It is a story that is no longer exclusive to archaeologists and anthropologists. It's a story for all of us because it belongs to all of us. And as more DNA is extracted, more cave floors sampled, and more interstellar data compared, We will continue uncovering new chapters, some that challenge our beliefs, others that affirm our deepest intuitions. We now know that the earliest Homo sapiens were not a single tribe, but a constellation of populations spread across Africa and beyond. Interbreeding with other hominins was not rare, it was common and often beneficial. Migration was not a one-way road out of Africa, but a complex web of movement in every direction. Ancient humans were artists, travelers, and storytellers, not primitive survivors, but cultural innovators. And perhaps most profoundly, that our past is not dead. It lives in our DNA. This understanding changes how we think about the present. It shifts our sense of identity. It reminds us that race is a social construct, not a biological destiny. That borders are recent, while migration is ancient. That the differences we see today are superficial, and the similarities run deep. In an age where division and isolationism are on the rise, the real story of human origins is a powerful counter-message. It tells us that collaboration, diversity, and openness were not just ideals. They were survival strategies. It tells us that our ancestors didn't build walls. They built bridges. As we look to the future, facing climate change, pandemics, and global migration, this ancestral wisdom could not be more relevant. Just as we once survived droughts, volcanic winters, and glacial advances by relying on each other, we must do the same again. Our story is still being written. Every DNA sequence, every archaeological dig, every celestial observation adds another sentence. But the theme remains the same. ***, we are one species, shaped by many histories. ***, to understand where we're going, we must understand where we came from. And thanks to ancient DNA, Atlas, and even the stars above us, we're finally beginning to see the whole picture. This is not the end of the human story. It's a new beginning. If this story made you rethink everything you thought you knew about human history, you're not alone. From bones to genomes, from Earth to the cosmos, our search for who we are has never been more complex or more inspiring. What we found wasn't a single origin, but 1,000 stories braided into one human journey. And the more we learn, the more we realize we're not just descendants of survivors. We're descendants of explorers, innovators, and connectors. If you want more deep dives into the origins of our species and the secrets still buried in our DNA, don't forget to like, subscribe, and hit that bell, because this journey, it's just getting started. For centuries, our understanding of the first humans relied heavily on a handful of fossil finds scattered across East Africa. Names like Omo Kibish, Jebel Irhoud, and Herdo Buri appeared in textbooks as the birthplace of Homo sapiens. Modern humans, round 100,000 to 300,000 years ago. But with the rise of ancient DNA analysis and global genome mapping, the picture has become far more complex, surprising, and in some ways humbling. We now know that there wasn't a single first human who emerged in one place at one time. Instead, what we call Homo sapiens likely rose from ***, a network of interlinked populations ***, spread across a vast geographic region that stretched from southern to northern Africa, and possibly into parts of the Middle East. In fact, some researchers now describe early Homo sapiens not as a species with a center, but as a *** species with a range ***, much like how modern animals occupy different terrains and evolve localized traits. Early human groups were genetically diverse, physically variable, and separated by vast distances. Yet they still interbred, shared technologies, and left similar archaeological footprints. The key word is ***, mosaic ***. Just like a mosaic is made of many small, unique tiles, early Homo sapiens were made up of genetic, anatomical, and behavioral elements inherited from different hominin groups. Some populations had features that looked more modern, rounded skulls, small jaws, while others retained archaic traits, and all of them contributed to the emergence of modern humanity. This new view is sometimes called the *** Pan-African model ***, the idea that Homo sapiens emerged through the interaction of various African populations, not from a single cradle of humanity. And yet, this model also expands beyond Africa. Thanks to ancient DNA, we know that Homo sapiens left Africa in multiple waves. The most well-documented one occurred around 60,000 years ago, when humans spread rapidly across Eurasia, but earlier migrations, dating to 120,000 or even 180,000 years ago, have also been found, especially in the Levant, China, and Southeast Asia. Some of these early pioneers left descendants. Others did not, but their presence reshapes our timeline dramatically. Even more compelling is the ** evidence of interbreeding **. As Homo sapiens moved through Eurasia, they encountered Neanderthals, and they didn't just fight or ignore them. They mated with them, multiple times, and in multiple places. The genetic legacy of Neanderthals exists today in every person of non-African descent, in traits related to immunity, skin tone, hair texture, and even neurological development. This means the first humans weren't pure Homo sapiens. They were ** hybrids **, biologically enriched by the species they encountered. In Southeast Asia, something similar happened with the Denisovans. Though we still know little about their appearance, we know their genetic influence is strongest in Melanesians, Aboriginal Australians, and some Southeast Asian groups. One Denisovan gene variant is responsible for high-altitude adaptation in Tibetans, a direct link between ancient encounters and modern survival. Perhaps the most surprising twist is the discovery of ** ghost lineages hominin populations, whose bones we've never found, but whose DNA survives in us. In sub-Saharan Africa, for instance, genomic data points to ancient admixture events with a mysterious group that may have split from our lineage over 1 million years ago. These ghost populations add new branches to our already tangled family tree. All of this leads to a profound realization. The first humans were not defined by a single trait, location, or moment. They were defined by ** connection **, by the constant exchange of genes, culture, and knowledge. They made tools, painted caves, buried their dead, and traveled immense distances long before cities or agriculture existed. They experimented, failed, adapted, and thrived. And in doing so, they became us. What's even more remarkable is how mobile these early humans were. Long before written language or wheeled transportation, our ancestors were navigating forests, deserts, mountains, and seas. Evidence from ancient tools and DNA suggests that humans reached Australia at least 65,000 years ago, likely by boat. This means they possessed not only the tools to survive, but the ability to imagine distant lands, plan journeys, and communicate ideas with precision. In the archaeological record, signs of creativity and abstract thinking begin appearing early. Red ochre pigments used for symbolic art have been found in South African caves, dating back 100,000 years. Carved beads, engraved bones, and painted shells show that early humans had a concept of identity and culture. This cultural explosion likely coincided with, or was supported by, genetic changes that enhanced cognition, memory, and social complexity. One theory proposes that this innovation was tied to the development of complex language, not just a set of basic signals, but full grammar, metaphor, and storytelling. Though language doesn't fossilize, the growth of brain regions associated with vocalization, combined with symbolic artifacts, points to language as one of the most powerful tools Homo sapiens ever developed. Ancient DNA has even allowed researchers to estimate when key genes for speech and hearing evolved. The FOXP2 gene, once thought to be uniquely human, has now been identified in Neanderthals as well, suggesting they, too, had some capacity for speech. All of these insights, from genetic hybridity to cultural expression, push back against the idea that early Homo sapiens were fully distinct from other hominins. Instead, we see a ** spectrum of humanness ** shared across different groups, with traits evolving in parallel or being exchanged through contact. Even the idea of extinction is being reconsidered. Neanderthals and Denisovans may have disappeared from the fossil record, but parts of them live on in billions of people. Their languages, rituals, or technologies may have influenced Homo sapiens directly. In this light, extinction isn't disappearance, it's absorption. Another key shift in our understanding involves how Homo sapiens adapted to the many environments they encountered. As groups moved into new territories, deserts, tundras, high-altitude mountains, they underwent natural selection for traits that helped them thrive. Ancient DNA has shown how gene variants for skin pigmentation, vitamin D metabolism, and even fat storage became more common in populations living in extreme climates. For example, populations in Northern Europe evolved lighter skin to better synthesize vitamin D in low sunlight conditions. Meanwhile, Inuit populations developed genes to efficiently metabolize omega-3 fats from their marine heavy diet. These adaptations didn't happen overnight. They were shaped by centuries of trial, error, and survival. The power of ancient DNA lies in its ability to show us these changes in real-time. By comparing genomes from different time periods and locations, researchers can trace the spread of beneficial genes, the collapse of genetic diversity during bottlenecks, and the reintroduction of traits through interbreeding. It also reveals the costs. Some genetic variants associated with past survival are linked to modern health conditions. The same genes that protected ancient humans from pathogens may now increase susceptibility to autoimmune diseases. Others may influence metabolism in ways that clash with modern diets. This evolutionary legacy reminds us that we are still evolving still carrying the marks of the paths our ancestors walked or were forced to walk. Another major insight from recent findings is the unexpected genetic diversity of early humans. For a long time, it was assumed that ancient human populations were small and isolated, leading to limited diversity. But DNA from early Homo sapiens fossils shows surprisingly high genetic variation, especially in Africa. This challenges older models that relied on simplistic bottlenecks or single origin points. Instead, it points to large, interconnected networks of communities, trading, mating, migrating, and exchanging ideas across vast distances. This paints a picture of ancient Africa not as a static landscape, but as a dynamic web of innovation, movement, and cultural fusion. The same goes for Asia, which has often been overlooked in discussions of early human evolution. New fossils and genomes from places like Israel, India, and China suggest that Asia may have hosted multiple waves of early Homo sapiens and Denisovan-like humans long before we thought. The out-of-Africa narrative is giving way to a more inclusive model of out of everywhere and back again. Genetic data also supports the idea of back migration that is, Homo sapiens groups who left Africa later returned, bringing new traits, technologies, and genes. This backflow helped shape African diversity and may explain some of the mixed traits seen in ancient African fossils. What's emerging is a story that resists easy categorization. It's not first came A, then B. It's more like A, B, and C existed at the same time. They met, merged, separated, and repeated the cycle. Evolution didn't move in a straight line. It wove itself into braids, branches, and feedback loops And at every turn, ancient DNA is helping us reconstruct the dance, step by step. The first humans, in the sense we once imagined, may never have existed. There was no single pair of individuals who gave rise to the modern species. Instead, there was a population, or better yet, populations that gradually accumulated the